right, diving in today. And we're going deep on something pretty fascinating, I think.
Yeah.
How our genes, our DNA, how that actually affects the way our brains age.
Yeah, definitely. A lot of people are interested in this topic. And there's this really interesting research article out of the UK Biobank that we're going to look at today. It's called Genetically Supported Targets and Drug Repurposing for Brain Aging, a Systematic Study. And it's basically trying to tease out, how our genes can make our brains age faster, slower. What's going on there?
Yeah, and I think the goal here for us is really to understand, are there any clues in our genes about how we could, keep our brains sharper, healthier as we get older?
Exactly, yeah. And a really important concept that they used in this study is the brain age gap.
Okay, brain age gap. So, is that just how old your brain is?
Well, so think of it this way. Your chronological age, that's just, how many years you've been alive. Straightforward, right?
Right.
But brain age, that's different. It's an estimate of how old your brain actually looks based on scans. Like in this study, they used MRI scans, and then they used these really fancy computer programs, deep learning models, to analyze the scans.
So the computer is looking at the scan and saying, oh, this brain looks like it's this old.
Exactly, yeah. And the difference between that estimated brain age from the scan and your actual age, that's the brain age gap. And that's what they're exploring as a potential sign of how healthy your brain is. So if your brain looks older on the scan than you actually are, that could be, a bit of a warning sign.
A little bit concerning, yeah. So, okay, how do they measure this? This brain age gap thing, it sounds like it involves a ton of data.
It did, yeah. They used the UK Biobank dataset, which is this huge, huge collection of health and genetic information. For this study, they specifically looked at MRI scans from over 38,000 people and genetic data from over 31,000. And the key here was that they trained these deep learning models. Remember those fancy computer programs? Mm-hmm. They trained them on the MRI data from healthy participants. So the models could learn what a typical brain should look like at different ages.
Oh, so it's like teaching a computer to look at a brain scan and be like, oh, this is what a 20-year-old brain looks like. This is what a 40-year-old brain looks like.
Exactly. It's like how those programs can recognize faces in photos. Now they're recognizing age in brain scans. And they actually trained not just one, but 7 different really sophisticated deep learning models.
Wow, 7.
And the one that performed the best, the most accurate, was called the 3D Vision Transformer, or 3D VIT.
3D VIT, okay, I'm writing that down. So they had this 3D VIT model that could, accurately guess the age of the brain from a scan. But to make sure that it was working right, they tested it on other data as well.
Yeah, for sure. They wanted to make sure it wasn't just picking up patterns that were specific to the UK Biobank data. So they validated it on 3 other independent data sets. One that focused on Alzheimer's disease, another one on Parkinson's, and then a general brain imaging data set.
Oh, so different types of brains, different conditions.
Exactly. And the fact that 3D VIT worked really well on these different data sets made them more confident that it was a reliable tool.
Yeah, that makes sense. You don't want your tool to only work in very specific situations. So, okay, they had this reliable way to measure the brain age gap. What were some of the first things they noticed?
Well, one of the first things, and this is kind of what we'd expect based on what we already know about brain health, was that people who are diagnosed with brain disorders, things like Alzheimer's disease or demyelination, that's damage to the coating around nerve fibers.
Oh, right.
And also schizophrenia, they tended to have a bigger brain age gap. It's almost as if the model was picking up on the changes in the brain that are linked to these conditions and seeing those changes as accelerated aging.
Wow. So the model's basically confirming what doctors see in the real world. That's pretty cool.
Mm-hmm.
What else did they find out about this brain age gap?
So they also found some links between certain lifestyle factors and a bigger brain age gap.
Okay, what kind of lifestyle factors?
Well, specifically, they found that being male, smoking, and having a really high or really low BMI, body mass index, those were all associated with having a brain that looked older than the person's actual age.
So, not healthy habits, basically.
Yeah, pretty much. And we already know those things are bad for your overall health. But this suggests they might also be speeding up the aging of your brain.
It's like aging your brain faster than the rest of your body.
Okay.
So did the model show them anything about, are there certain parts of the brain that age faster than others?
Yeah, so they use something called saliency maps.
Saliency maps, that sounds complicated.
It's not that bad, actually. Think of it like a highlighter, highlighting the areas of the brain that the AI is paying the most attention to when it's trying to predict age. And what they found was that two areas in particular, the lentiform nucleus and the posterior limb of the internal capsule, those seem to be really important for estimating age.
Okay, I'm not going to pretend I know what those are, but are those areas, like, more affected by aging?
It seems that way, yeah. The lentiform nucleus is involved in things like motor control and movement, and the posterior limb of the internal capsule, that's a big pathway for nerve fibers. They also saw some weaker correlations with other areas like the thalamus, the corpus callosum, and the caudate nucleus.
Got it. So it's not just the whole brain aging uniformly. Some parts seem to be more, I guess, sensitive to age. All right, so we talked about this brain age gap and how it's linked to some disorders and lifestyle choices. Now, let's get to the genetics part. What did they learn about how our genes affect all this?
So this is where we dive into our DNA. They did something called genome-wide association studies, GWAS, on the brain age gap in those healthy people from the UK Biobank.
GWAS. So they're looking at the whole genome, like all the genes.
Yeah, basically they're scanning the whole genome to see if there are any variations in the DNA, like little changes in the letters of the genetic code that are associated with a bigger or smaller brain age gap.
So like looking for the genes that make your brain age faster or slower.
Exactly.
And did they find anything?
They did. They identified 9 distinct little changes, these single-letter changes in the DNA, in 8 different spots in the genome that were significantly associated with brain age gap. What's cool is they not only confirmed some genes that we already knew were involved in brain aging, like RUNX2, INP5A, CRHR1, and PICK1.
Okay, yeah, I've heard of some of those, at least I think I have.
But they also found some new ones, like TP53 and NKX2 too.
Oh, so new potential players in the brain aging game.
Yeah, exactly. And they found that one region in particular, called 17q21.31, had a really strong link to brain age gap.
17q21.31, is that like a chromosome number or something?
It's a specific spot on a chromosome, yeah. And in that region, there are some interesting genes. One is called CRHR1, and we know it's involved in how brain cells lose their connections with each other, which is called synaptic loss, and also in memory function.
So things that are, very important for a healthy brain.
Absolutely. And another gene in that region is NSF, which has been linked to Alzheimer's disease. So this suggests that variations in the genes in this region could really influence how your brain ages and maybe even make you more susceptible to age-related conditions.
Like a genetic predisposition to certain types of brain aging.
Potentially, yeah. But it's important to remember that genes aren't the whole story. They estimated that the heritability of brain age gap is about 0.2.
0.2, what does that mean?
So heritability is basically how much of the variation we see in a trait, like brain age gap in a population, is due to genetic differences. So 0.2 means that about 20% of the differences in brain age gap we see between people are because of their genes.
Okay, so genes matter, but they're not the only thing.
Exactly. The other 80% is probably due to things like environment, lifestyle, things that aren't directly encoded in your DNA.
So nature and nurture both playing a role. Now, they also wanted to see if this brain age gap had any direct links to other health problems, right? Did they find any?
So they looked at this really carefully, and after doing all the statistical adjustments, they found that there weren't many strong causal connections between brain age gap and 18 different brain disorders, or 8 other traits.
Wait, so even though people with those conditions often have a bigger brain age gap, it doesn't necessarily mean that the gap itself is causing the problem.
Right. It's more complicated than that. There could be other factors at play. The main exception they found was a possible causal link between brain age gap and intelligence.
So a bigger gap might be contributing to lower intelligence, but it's not clear if it directly causes things like Alzheimer's or stroke.
Exactly. It's a subtle but important distinction.
So, okay, they've found these genes linked to brain aging. What's next? Did they look at whether any of these genes could be targeted with drugs?
Yeah, that was their next goal. They wanted to see if any of these genes could be potential targets for treatments, to slow down brain aging or prevent problems. And to do this, they combined their G-Jeserways data with information about how genes are actually expressed, how active they are. That's called EQTL data. and also data on protein levels, pQTL data.
Right, because genes make proteins and proteins do the actual work in the body.
Exactly. So they wanted to find genes where the genetic variations that affected their activity were also the ones associated with brain age gap. Basically, they were looking for druggable genes, genes that we might be able to target with medication that have a causal link to brain age gap.
So find the genes, find the drugs that can affect those genes, and maybe slow down brain aging. How did they narrow down the list of potential targets?
They used a couple of powerful statistical techniques. One is called Mendelian randomization, or MMR, and the other is colocalization analysis. Mendelian randomization is a really clever way to figure out causal relationships using genetic variations as stand-ins. They found 64 druggable genes where the level of activity, influenced by genetics, seemed to have a causal link to brain age gap. Then they used colocalization analysis to refine that list even further to 22 genes.
So they're like double checking their results, making sure they're really onto something.
Yeah, they're being very thorough. And with colocalization analysis, they were looking for places where the genetic variations affecting gene expression and the variations affecting brain age gap overlapped. That overlap is a strong hint that there's a shared cause.
So it's not just random, there's a real connection there. And they also looked at protein levels, right? And make sure it wasn't just the gene activity, but the actual protein being produced.
Yeah, they wanted to confirm that it was all connecting at the protein level because a lot of drugs actually target proteins directly. And using pQTL data from some big studies, they found 6 proteins that had a significant causal effect on brain age gap and another 14 that had strong evidence of colocalization with brain age gap.
So looking at the proteins is like a validation step, making sure everything lines up. And after all this, they found some key genes that look really promising.
They did. Based on having strong evidence from at least 3 different types of genetic data, they narrowed it down to 7 key druggable genes: MAPT, TNFSF12, GZMB, SRPB1, GNLY, NMB, and C1RL. Those are the ones they think are the best targets for future research.
7 genes. Okay. I've definitely heard of MAPT before in the context of Alzheimer's disease.
Hey.
What do we know about these top genes and why they might be important for brain aging?
You're right, MAPT is a big one. It's really important for the structure and stability of brain cells, and when it goes wrong, it's a hallmark of Alzheimer's and other neurodegenerative diseases. The other genes have different roles. TNFSF12 is involved in inflammation and blood vessel health. GZIMB and GNLY are related to immune function. ARPB1 regulates immune cells. NMB is a signaling molecule involved in lots of different processes. And C1RL is part of the immune response. It's really interesting that genes with such different functions could all be involved in brain aging.
Yeah, it's like a whole system, not just one thing. So after identifying these targets, they wanted to see if they had effects beyond just the brain.
Right. They didn't want to just focus on the brain in isolation. So they did something called a phenomoid scan, which is basically looking at how these genes are connected to other traits and conditions. They looked at 44 different traits like blood pressure, BMI, smoking habits, asthma, a whole range of things. They wanted to see if targeting these genes might have effects on the whole body, not just the brain.
And what did they find?
They found that these genes weren't just linked to brain aging. They were also associated with lots of other things like blood pressure, BMI, smoking, asthma, even neurodegenerative and psychiatric disorders, and various biomarkers. For example, they found that MAPT activity was linked to glucose levels and Parkinson's disease risk, SIRPB1 to insomnia and inflammation, and C1RL to OCD and Alzheimer's risk.
Wow, so these genes are really multitaskers. And that's important to know because a drug that targets one of these genes to slow brain aging might have unintended consequences elsewhere in the body.
Exactly. It's a complex web of connections.
So the last part of their research was looking at existing drugs.
Right.
Did they find any that already target these genes?
Yeah, this is where the idea of drug repurposing comes in, using existing drugs for new purposes. They found 29 drugs that have shown some promise in clinical trials related to aging, and that target 29 of the 64 genes they identified.
So some of these drugs are already being tested for age-related conditions.
Yeah, 13 of them are in clinical trials for various aging-related conditions. Some of them are things you might have heard of, like vitamin D3, dasatinib, diclofenac, omega-3 fatty acids, estrogen, hydrocortisone, mecamylamine, nicotine, prasterone, quercetin, resveratrol, sirolimus, and testosterone.
Wow, that's quite a list.
It is. And that's exciting because it means these drugs might be good candidates for further research specifically on their effects on brain aging.
Yeah, it could be a much faster way to develop treatments if we can use drugs that are already out there and have some safety data. They also pointed out some drugs that weren't typically thought of as anti-aging drugs.
Yeah, they found another 28 drugs that target genes like MPL, CA4, TUBB, and RRM1, and these drugs have mechanisms of action that could potentially be beneficial for slowing brain aging, even though they're not currently classified as anti-aging drugs. For example, drugs that activate MPL might increase platelet count, and there's some evidence that platelets might have rejuvenating effects on the aging brain. They also highlighted drugs that inhibit CA4, RRM1, and TBB as potentially useful.
So this research really opens up a lot of possibilities for future treatments, both with existing drugs and with new ones that could be developed to target these key genes. So let's zoom out a bit. What are the main takeaways from this deep dive into this study?
Well, I think this study gives us a really comprehensive look at the genetics of brain aging. By combining advanced brain imaging analysis with the huge amount of genetic data, they've pinpointed specific regions of the genome and, crucially, 7 really promising genes that seem to be strongly influencing how our brains age. Those genes, remember, are MAPT, TNF SF12, GZMB, SIRRPB1, GNLY, NMB, and C1RL. This gives us some really valuable targets for developing new treatments.
And what does this mean for future research and potential treatments?
These findings give us a strong foundation to build on. Now we can focus our research on developing therapies that specifically target these genes with the goal of slowing down or even preventing age-related cognitive decline. And the drug repurposing angle is especially exciting because we could potentially see treatments develop much faster if we can use drugs that are already out there.
But it's important to remember that every study has limitations, right? What were some of the things that we should keep in mind about this research?
Yeah, for sure. First of all, the models they use to estimate brain age, while they're very sophisticated, they're still just estimates. They're not perfect. And this study was mainly done with people of European white ancestry, so we need to do similar research in other populations to see if the findings hold up across different genetic backgrounds.
Yeah, we always need more diversity in research.
Absolutely. And while they did validate their model on different data sets, it would be great to replicate the genetic findings in a completely separate group of people just to be extra sure. And it's important to remember that brain aging is incredibly complex. This study couldn't capture all the factors involved. And lastly, even though they identified drugs that might be useful, they didn't look specifically at potential side effects, which is something we need to consider very carefully.
Right, so there's still a lot more to learn. Okay, as we wrap up this deep dive, what's a final thought you'd like to leave our listeners with?
Well, I think this research opens up some really exciting possibilities. We're starting to understand the genetic basis of brain aging, and that could lead to completely new ways to protect our brains and keep them healthy for longer. Imagine a future where we can take personalized steps based on our individual genetic makeup to keep our minds sharp and vibrant as we age. That's the kind of future this research is pointing towards.
That's an incredible thought. Thanks for taking this deep dive with us today.
It was my pleasure.
And to all our listeners out there, keep those brains active, and we'll catch you on the next deep dive.